is Kiera Blawn, and I study migraines and its relationship with OATP. So first of all, what's the difference between a migraine and a headache? So headaches typically occur with head pains or over pretty fast, whereas migraines have other symptoms as well as the pain, like nausea and dizziness, loss of vision and other sensory disturbances, and they can last several days. So this can actually have an economic toll on patients if they have to be hospitalized and miss several days of work. So this is obviously a huge burden to patients, and in addition to being a huge burden, a lot of people suffer with migraines, so around 1 billion people worldwide. And so it's such a prevalent neurological disorder, it makes it really important to study and find effective treatments. So my lab actually studies the basic mechanism of migraines. So we want to know what's causing migraines, what's going on, and what's changing in the brain during migraine, and that way we can target more effective treatments if we know what's causing them in the first place. So it can also teach us a lot about the brain in general and what happens when things go wrong. So we study the phase of migraine called aura. So aura precedes the migraine attack, and it usually includes sensory and visual disturbances. So sometimes people have loss of vision, or they can maybe see weird things in their visual field. They can also have other disturbances such as like sensitivity to light and sound. So people with aura know that their headache is coming because they have this phase and this disruption, and so they can kind of predict the migraine attack. And we study aura because of the unique electrical event that happens during it called cortical spreading depression, or CSD. So basically, this is an electrical event that we've noticed that happens where there is a wave of hyperactivity followed by inhibition. So basically what that means is the neurons depolarize and fire, and then they stop firing for a period of time. And this inhibition or depression spreads across the cortex from the occipital lobe, which could explain those visual problems, all the way up unilaterally, so on one side of your brain. And this is kind of an example of what an electrical signal would look like from those neurons. So in panel A, we see that the neurons were stimulated and fired, and then they stop firing for a period of time before they can fire again. And so cortical spreading depression is a super interesting phenomenon, but we can study it in an animal model and confirm that the animal is having similar electrical events in their brain that humans do during migraines. And so this makes it easy to quantify and measure that we are causing the migraine in animals. So we specifically look at the blood-brain barrier during cortical spreading depression. So the blood-brain barrier are basically specialized blood vessels. So in a normal blood vessel, we have endothelial cells that are packed together, and they keep the blood in, and they let materials through to the body tissues and back in. But in your brain, you have very sensitive neurons, and so it's important that we protect those neurons and only let things through to the brain that should go to the brain. And that means that the endothelial cells in these blood vessels in the brain are packed much more tightly together. So they have 100 times more tight junction proteins, and then we also have these glial support cells called astrocytes that surround the blood vessel and form an even tighter seal. So basically what that means is things aren't going to be able to go through cells, between cells like they are in normal blood vessels to get to the brain. So they either have to be lipophilic to go through the membranes into the brain, or they have to have a specialized transporter. So the analogy I like to use is TSA. So in security, people want to get through security to their gate, but they can't go in between the stations. They have to go through security checkpoints. And so this is the same with molecules in the blood that want to get to the brain. They have to go through transporters. And the people that go through security, they have to meet specific qualifications. So for example, having a boarding pass or going through a metal detector. Just like brain molecules in the blood that want to get to the brain, they have to meet that transporter's specific qualifications. So transporters are specific for certain substances. And so only substances that have a transporter are going to get through the brain, just like only people with boarding passes are going to get through their gate. So in cortical spreading depression, what we see with the blood-brain barrier is that it has an increase in permeability. More things are getting through to the brain. And so that would be maybe analogous to having more security checkpoints, so more people can get through to their gates. So what we think is that there's an increase in transporters, and that's what's causing this change in permeability, and more things are getting through. So we decided to look at the transporter OATP, which is organic anion transporting polypeptide. Basically what that means is it transports organic or carbon-based molecules that have a negative charge. So this is an example. We have a carbon skeleton up here, and then this negative phosphate group. So a popular anti-migraine drug called sumatriptan has a similar structure. It's got this carbon skeleton and this negative sulfate group here. So we think that OATP can transport sumatriptan into the brain, and that's how it helps migraines. So if we're seeing an increase in transport during cortical spreading depression, then maybe there's a better phase of the migraine to deliver this treatment to. And so how do we study OATP? We can do it in two ways. We can do it in our in vivo rat KCL animal model, or we can do it in our cell lines, which is in vitro. So in the rat model, we basically do surgeries so that we can inject or insert a cannula and inject KCL into these rats. And so a potassium chloride solution basically causes a migraine in rats, and that's our migraine model in the animals. And so how do we know a rat is having a migraine? Well, a couple of ways. We can see that same electrical event that I talked about, cortical spreading depression, in these rats. And then we can also do behavior, and we see similar pain responses in the rats that we see in humans. So facial pain aldonia, which is pain when you touch right between the eyes. We can see that in humans with migraines and also in rats. So that is one way we can study migraines and OATP in rats. But then we can also go and control for smaller changes in the cell level. So we have those astrocyte cells, which I talked about, surround the blood vessel and the blood-brain barrier. And we also have the BEN3 cells, which are endothelial cells of the blood-brain barrier. And so we can go and, like, for example, knock out or inhibit OATP in those cells, and then put radioactive sumatriptan, that anti-migraine drug I mentioned, and we can measure directly the transport. And so we can see if OATP is directly transporting OATP using the cells. So that's what I'm working on, and I'd just like to acknowledge and thank both the labs that I work with and all of my funders. Thank you.